phospholipids. That is a glycerol, a 3-carbon glycerol, so a C, a C, a C, a carbon, a carbon, a carbon, and to 2 of these carbons there's a fatty acid tail. So a fatty acid at 1 carbon and another one, but then at the 3rd one there is then a little phosphate group, PO4-3 is written in the book, and at that then there is then a little other positive group containing oftentimes nitrogen. And that is now the positive part, and that can make hydrogen bonds with water. By a hydrogen bond, for example, by a hydrogen bond with water between the oxygen and the 2 hydrogens, where the oxygen is a heavier atom and it pulls stronger at the shared, in a covalent bond, shared hydrogen as the little hydrogen is just pulling at it. And so the electron is a bit pulled away from the hydrogen, and the hydrogen has lost a bit its negative electron, and the hydrogen is a bit more positive. The book leaves an open why now that positive hydrogen can team up with the positive part of phospholipid head, whatever. Now, so a phospholipid, 3-carbon glycerol, carbon, 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 and 2 of them fatty acid tails, and at the other one there is a little phosphate group, and then something with polar positive head containing oftentimes nitrogen. And they are at the cell membrane of cells, and the positive polar head turns to the water. It is hydrophilic, it allows the water to the extracellular fluid at the outside, and another row of them turns to the other side, to the cytosol, the fluid part of the cell on the inside. So they're standing then like that, and on the inside are the fatty acid tails, and then there's a polar head going to the cytosol and to the extracellular fluid. And these fatty acid tails, which can make bonds with lipids, they have oftentimes between all these carbons, when in the fatty acid tails, a double covalent bond. And then they are not together with one bond, but at that one place they are then double together, and that gives it then a little bend. And therefore they are oftentimes like that shown. And that is according to Gerard and Senegafovsky's Principles of Anatomy and Physiology book on the pages 46 and 47. But this is no medical advice and also no substitute for medical knowledge. The positions and views expressed are the ones of source and don't necessarily align with my own or with the ones of the entity spoken of. Hi and welcome. I am trying to learn biochemistry. Today, phospholipids. After we've done that, we're going to do tiptoe yoga. And if you participate in the tiptoe yoga, then it's better to ask. Phospholipids, like triglycerides, phospholipids have a glycerol backbone and 2 fatty acid chains attached to the first 2 carbons. A glycerol backbone, and then 3 fatty acid tails, 2 fatty acid tails to it, and in the 3rd position, however, a phosphate group links then a small charged group that usually contains nitrogen to the backbone. So we're having here the 3-carbon triglyceride glycerol, and at the first 2 there are fatty acid tails, and then we're having here the phosphate group, to which then the nitrogen group is added. It contains nitrogen. It's just a polar head containing the nitrogen added through the phosphate group to the 3rd carbon of a 3-carbon triglycerol. In the 3rd position, however, a phosphate group links PO4-, links a small charged group that usually contains nitrogen to the backbone. This portion of the molecule, which is called the head, is polar and can form hydrogen bonds with water molecules. A phospholipid has one of a 3-carbon glycerol backbone. It's not a triglyceride backbone. Has it a phosphate group to which has it a phosphate group links then a small charged group, nothing else, a small charged group which oftentimes contains nitrogen. And that thing is polar and can form hydrogen bonds with water molecules. Hydrogen bonds, so it can go to water. How would it be now with nitrogen? It is H3C-. Hydrogen bonds, there is when always the hydrogen share is a bit stronger, or by the other side, it was it? Where was it said? The polar covalent bonds that form between hydrogen atoms and other atoms can give rise to hydrogen bonds. Hydrogen and other atoms. This type of chemical bond, 27. The polar covalent bond causes the hydrogen atom to have a partial positive charge. It's a polar covalent bond. Why was it polar? The sharing of electrons between two atoms is unequal. One atom attracts a shared electron more strongly because the oxygen is stronger pulling at it as the hydrogen. Or, yes, the hydrogen is bonding up with water. And the oxygen is pulling stronger at the shared electron as the hydrogen. And therefore the hydrogen is a little bit more positive because the electron is stronger put into the oxygen, and it has less electron, and therefore it is positive. And it is now hydrogen bond between the hydrogen and the oxygen. And now the phosphate can do that as well, do I say? This portion of a molecule is polar and can form hydrogen bonds with water molecules. Whatever, it can make hydrogen bonds with water molecules. With water molecules. So the nitrogen would be pulling stronger at the hydrogen, I think, of the water. Or if the nitrogen is positive, the nitrogen is positive and the water is, the hydrogen is also positive. Can it then bound to the oxygen? The two fatty acids, the tails, on the other hand, are non-polar and can interact only with other lipids. I don't think we're going to find it out. What is happening now exactly with the phosphates? Who is now at what sharing and where exactly the polar heads are happening? So we're just going to say it as it is happening here. That is it is polar and it can make then hydrogen bonds with water molecules. The hydrogen bonds where the one electron of a hydrogen, where the electron makes is it also a covalent bond? It is also a covalent bond. A covalent bond. I think all hydrogen bonds were covalent bonds, but not all covalent bonds hydrogen bonds. Neither of the combining atoms loses against electrons. Yes, hydrogen bond is the polar covalent bond. And polar is when it's unequal pulled at it. And the hydrogen bonds are the ones resulting from it when with the hydrogen. The polar covalent bond causes the hydrogen atom to have a partial positive charge that attracts a partial negative charge of neighboring electronegative atoms, most often oxygen or nitrogen. So nitrogen is negative for whatever reason. Hydrogen bonds are weak. So the hydrogen bond is not the hydrogen with the oxygen, that is the polar covalent bond. The hydrogen bond happens when with the hydrogen with the nitrogen. And that could happen here. The portion of the head polar can form hydrogen bonds with water molecules. And that nitrogen can form them with polar bonds because nitrogen is negative and the hydrogen is positive because the electrons of the hydrogen was pulled to the oxygen of the water. The two fatty acids, the tails on the other hand, are non-polar and can interact only with other lipids. Why are they now non-polar? In some covalent bonds, two atoms share the electrons equally. Yes, these are non-polar. Polar was that the oxygen is pulling stronger at hydrogen. One atom does not attract the shared electron more strongly than the other atom. This type of bond is a non-polar covalent bond. The bonds between two identical atoms are always non-polar covalent bonds. Why is now a fatty acid tail a non-polar one? It's all the time carbon and with no hydrogen. It's non-polar. Molecules that have both polar and non-polar portions are said to be amphipathic. And a phospholipid is such a one. Phospholipids, which have this unusual property, line up tails to tails in a double row to make up much of the membrane that surrounds each cell. And molecules of which a part is positive and another one, of which a part is polar and another one non-polar, are called amphipathic. One part of them attracts water and another one repels it. And the phosphate head with the nitrogen in it wants to make, because the nitrogen is negative, wants to make with a slightly positive hydrogen, which is in a polar covalent bond with oxygen together in a water molecule coming together, and the nonpolar for some reason don't want to. Nonpolar, why are we nonpolar? Because it's always carbon with carbon. And the outsides of cells have it all the time. The cell membrane consists out of them where, like, the phosphate head at the top. And why is the phosphate head so much at the top? Phospholipids. In the synthesis of phospholipids, two fatty acids attached to the first two carbons of glycerol backbone. A phosphate group links a small charged group to the third carbon in glycerol. And then there is a phosphate group consisting PO4, 3 P, O4, and 3 minus. We're having a phosphate in the center, and then 4 oxygen around it, a phosphate group. PO, so 1 phosphate, 1 P, and then 4 times VO, 4 times oxygen, here 4 times oxygen. And then we're having 3 minus. Why do we have 3 minus? There's once a double covalent bond between phosphate and oxygen, and when we're having then oxygen with a minus written next to it. A phosphate group links a small charged group with a third carbon in glycerol. In B, the circle represents the polar head region, and the two wavy lines represent the two non-polar tails. Double bonds in the fatty acid hydrocarbon chain often form kinks in the tail. And they are making with these lines because they say we're having these kinks there, these bends, because at some places, for whatever reason, why is it there? For some reason, their carbon has not a single bond with another carbon, but 2 electrons are shared, because the electrons are all proportionally around the atom, at the outside. They can't just meet perfectly like that with the second one. The second one is further out there, they're all going around like that, so they have to go over to form to the second one. Okay, we have it together, but I'd like to make it properly. Let's read for one moment into steroids if there's something written, which is part of phospholipids. Steroids have 4 rings of carbon atoms designated A, B, C, and D. Their structure differs considerably from that of the triglycerides. Our steroids may be synthesized from cholesterol, which has a large nonpolar region consisting of 4 rings and a hydrocarbon tail. In the body, the commonly encountered steroids such as cholesterol, sex hormones, cortisol, bile salts, and vitamin D are further classified as steroids because we have a hydroxyl alcohol group -OH attached to one or more of the rings. These polar hydroxyl groups make sterols weakly apathetic. Although cholesterol can contribute to the buildup of fatty plaque in atherosclerosis, it also serves important functions as a component of animal cell membranes and as the starting material for the synthesis of other steroids. Okay, now to the phospholipids. That means if you were here for biochemistry, you can go directly to the next part, where we try to make our little presentation and say these few things that were said over the phospholipids that we have them in a little presentation. Later on, we're going to do the steroids as well as the steroids. If you stay here and participate in the tiptoe yoga, then it's very honest. Have feet standing together, breathe in the arms up, come up to the tiptoes. Out and down again. Come always to the big toes with the weight. In and up. Out and down. In and up. Out and up to the side. In and up. Out and up to the other side. In and up. Make an inch of space between the feet, out and to the side. In and up. Out and down. In and up. Out and up to the other side. In and up. And out and down. In and up. And out and lean onto the knees. You can go with the heels down. Breathe in. And out, make a health of the back. Breathe in. And out, make a belly of the back. In middle. Out the head. In middle. Out the belly. Middle. And out, come with the round back. Up at the top of the back. Shoulder roll. Heels down. Breathe in the arms up onto the toes. And breathe out and sink down. This can go onto the knees. In and up again. out and down, in and up, out and hands down, heels down. Legs to the sides, 1 foot faces in front, the other one to the side. The heels are on a line, in the arms up. And out bend the front leg and come with the front foot onto tip down. Breathe in, reach in front, and out and back. Breathe in and straighten the front leg. Out, bend down, middle, down with the heel. Change the position of the feet, breathe in. Out, bend the front leg. And come over the tip down. In, reach in front. Out, lean back. In, straighten the front leg. Out and down again. Middle, down. The feet apart, in the arms up, onto tiptoe. Breathe out, sink down, and breathe in, and push something imaginary heavy upwards, out and down. In and up. Out and down. In and up. Out, hands down, heels down. And have the feet centered together, in the arms up onto tiptoe. And out and down, heels down. Bend the right leg, stretch the left one. Change and stretch the right one. Middle, both legs at the same time. And out the feet to the side. Keep on hanging down. Breathe in. And out, go over to one leg. Breathe in. Out and go over to the other leg. Breathe in. Out into the middle. And close the feet. Breathe in. And out. Keep on hanging down. Again in. And out comes slowly with the round back up and against the top and backwards. Shoulders down. Toes are pointing to the no, feet are staying to go. In the arms up. And out and down just. That was the tiptoe yoga. I thank you very much and until next time.